of the algae, so if it's the low temperature factor or the low pH, which cause the highest changes. And I want to determine changes in the photosynthetic and calcification rate from the algae which I have transplanted to the west and the one which remain in the east. And therefore also I incubate them at different pH and different temperature conditions. Yeah. Now I want to explain my experimental design. At first, the transect diving to know the distribution of Hallimeda. I worked in Yai, and we found 4 stations where I did transects. So 2 in the east, in the sheltered side, and 2 in the west, in the wave exposed side. And each station we had 3 different depths. So we did transects in 3 different depths, in 5 meter, 10 and 15 meters. And in each depth we did 2 replicates of 100 meter transects. On the right side you can see the transect line, and we counted Hallimeda numbers, the number of Hallimeda, and we measured the size 1 meter left and 1 meter right from the transect line. I cannot show you any results of the transect diving yet, because of course I have no maps yet, but we finished now with all the transects, or nearly all the transects, and at the moment it looks like that Hallimeda only occurs in the east side, so only in the sheltered side of the island. Doing the transects, as I said before, we counted the number and the size of Hallimeda and the sea bottom. So we monitored if Hallimeda is growing on sand, on rubble, on rock, on living coral or healthy coral, and on these dead coral fields which occur because of the tsunami in Yai. I don't know if you've seen them before. My second experiment, or the second part of my experiments, is an incubation experiment. I did this incubation experiment in January and March. I had an incubation and a transplantation experiment, so in January in the low soliton season, so when the waves do not occur that often, I took some Hallimeda from the east side, bring them to the boat, and then we transplanted them, we put them onto racks and transplanted them to the west side, so to the wave exposed side. And I took some Hallimeda to the lab here in the PMBC and did some incubation experiment. In March, so 2 months later, I did the same incubation experiment again with the algae I've transplanted to the west and the ones which I've transplanted to the east, so with both sides. Here are some pictures from the transplantation. This is my algae, which I've glued to these acrylic plates. The plates we fixed onto racks, and the racks we then transplanted into the reef onto the sandy bottom in 10 meter depth. My incubation experiment in January, I took out Hallimeda just from the east side, and I incubated them at 4 different treatments. I had a control treatment where the temperature and the pH are as in normal seawater, so I have a temperature of 29 degrees and a pH of 8.2. My second treatment is a low pH treatment where the pH is reduced to 7.7. Then I incubated at low temperature where the temperature is reduced to 20 degrees. And I have a large amplitudal internal wave treatment in which both factors are reduced. So I have a temperature of 20 degrees and a pH of 7.7. During my incubations, I put Hallimeda in these glass chambers for 2 hours, and in light and dark conditions. And I did always 6 replicates and with 3 blanks. So blanks are just empty chambers just filled with normal seawater to see how the seawater itself changes. You can see one incubation chamber with Hallimeda, and on the left side you can see the pump fluorometer with which I can measure the photosynthetic performance of the algae. As well, I monitor the pH, the temperature, and the oxygen. At the beginning and the end of my incubation, I measure, or I can calculate the calcification and decalcification of the algae in these 2 hours. The oxygen production and consumption, the photosynthetic performance, and the chlorophyll content. Yeah. Now I want to show you my first results from January, from the incubations in January. At first, it's a graph about the photosynthetic performance. So on the x-axis you can see the beginning and the end of the incubation, so the incubation time. And on the y-axis you can see the photosynthetic yield. I don't know if you know what it is, but the yield shows the higher or you can say the higher the yield, the better is the photosynthetic performance. And yeah, the black line you can see is a control treatment in which the photosynthetic is quite stable during the incubation, while the photosynthetic rate is reduced in low pH conditions, in low temperature and the large amplitudal internal waves. Yeah, the photosynthetic rate is much lower than the control condition. So when you look at the photosynthetic rate, it seems that the low temperature has the low temperature and the combination of reduced temperature and pH has the greatest effect on Hallimeda. However, my oxygen results show different pattern. On the upper graph you can see the oxygen production per fresh weight per hour, and on the lower graph you can see the oxygen consumption during 1 hour. And there it seems that the photosynthesis is highest at low pH. So when I incubate them at a pH from 7.7, they show the highest oxygen production, while the oxygen consumption is quite stable between all treatments. Yeah, why the photosynthetic rate and the oxygen production is so different, I don't know yet, but I'm sure that I will find a reason. Yeah, I also calculated the calcification rate, and there you can see that in the control treatment, so at normal seawater conditions, the algae calcify a lot. They have the highest calcification. During the large amplitudal internal waves, so when the pH and the temperature is reduced, the calcification is lowest. So as I expected it before. You can also see, which is very interesting, that during the large amplitudal internal wave treatment, that when you calculate the net calcification, so the production minus the consumption, yeah, that you have decalcification in general and not a calcification. And this is different, for example, in the control treatment where you have a net calcification. The same incubation experiments I did again in March. With the algae I have transplanted to the west and to the east side of the island. But because I don't have enough fragments of Hallimeda, and I incubated them just at 2 treatments, so at the control treatment and the large amplitudal internal wave treatment. So with normal seawater conditions and the conditions where temperature and pH is reduced. I also incubated for 2 hours in light and dark conditions, and again I measured the calcification, the oxygen production, and the photosynthetic performance. The photosynthetic performance of these 3 incubations are interesting. I have, this is again the photosynthetic yield I explained before. And these are the mean yields I've measured for our algae from the January incubation and from the March East incubation and the Mars West incubation. And there you can see that the algae which I have transplanted to the west side, so to the wave exposed side, behave or have a higher photosynthetic performance than the one I've transplanted to the east. So that is quite different from what I have expected. The same pattern you can see in the oxygen production. The you have the oxygen production on the y-axis, on the x-axis the two different treatments. The black bar is the incubation in January. The one in the middle, the gray one, is the incubation in March. And the dark gray one on the right side is the incubation of the west algae in March. And there you can see also that the west algae behave better than the one in the east. So they have a higher oxygen production, while the oxygen consumption is quite stable during the incubation. I also measured the calcification again. There the pattern is different again. So during the large amplitude internal wave treatment, so when temperature and pH are reduced, I have calcification which is quite the same for all algae, for the one in January, for the one in March and from the west side, and the one from the east side, while the decalcification is reduced at the ones from the west side. So the shellfish algae have a higher decalcification than the one from the east side. So again, the east algae behave better than the west algae behave better than the east algae. Yeah, now I want to give you a very short conclusion, as far as I can say it yet, because I have not all results yet and not all data. But from the transect diving, I can say yes, Hallimeda occurs only at the east side of Puvacharyai, as I expected before. If this distribution pattern is influenced by large amplitudal internal waves, I don't know yet, but I can say that the fragments I've transplanted to the west side, so to the wave exposed side, behave better in general than the one which remained at the east side. So it seems that large amplitudal waves are not the only reason for the distribution pattern of Hallimeda opuntia. So there must be, or I think, as far as I can say it yet, that there must be another reason for the distribution of Hallimeda that is not occurring in the West. And yeah, thank you very much.